So welcome everyone to our first ever webinar on brain donation within our cerebral amyloid angiopathy research program here at Mass General Hospital. It is really a privilege to host this session for you. I think the reason that we decided to organize this informational webinar is actually twofold. I think first of all, we would really like to just say thank you to the families who have participated in this program in the past. Really, by donating the brain of a loved one, which is a really courageous thing to do, you have truly made such a precious gift and a much needed contribution to our research. And we just wanted to take the opportunity to say thank you for that gift. And secondly, we also imagine that you may wonder if anything has come out of these brain donations. And we couldn't actually answer that question more positively. Our research team has learned so much about the neuropathology and also the biology of CAA. And we would just love to spend some time giving you an idea of what we have learned so far. Just a few housekeeping rules before we really dive into the science. We are using the Zoom webinar format, which means that we cannot see or hear you. On your screen, you should, I think, see three options for communication. The first is a chat function, there is a Q&A function, and there is a raise your hand function. We kindly ask you to only use the Q&A button. And when you have a question, you can put them in there. You're encouraged to ask as many questions as you have throughout the webinar, and the panel will try to answer some of them during the second half. There is also a checkbox in there that you can tick if you'd like to answer or to ask your question anonymously. And that's also a possibility there. So as mentioned, this webinar is being recorded, and we will post a link on our various online platforms for you to listen to later or to share with your family and friends. We will also send a follow-up email to those who have registered for this webinar with contact information and a link to a survey to collect feedback and also ideas for potential future webinars. So I will share the presentation that we've prepared. Let's see. There we go. So just a quick heads up, the PowerPoint presentation does include some examples of MRI scans as well as microscopic images of the brain and a couple photographs, and that are obviously all fully de-identified. But we just wanted to mention this upfront so that you are aware of what you can expect and that you're not caught off guard. So this is the schedule of the webinar. We will first hear from Dr. Steven Greenberg, who is a professor of neurology at Harvard Medical School and Mass General Hospital, and the director of our hemorrhagic stroke research program. He will start us off by giving a brief overview of CAA and also introduce the brain donation program. Next up is Dr. Matthew Frush, who is a professor of neuropathology, also here at HMS of Harvard Medical School and Mass General Hospital, and the director of the autopsy service here at our hospital. He will discuss some findings on CAA and in the brain at autopsy. Then I will present a few slides on the research that we have been able to do using these brain donations. As you already guessed, my name is Dr. Susanne van Veluwe, and I am a neuroscientist. And my lab at MGH studies the biology of CAA, both in experimental models in the lab, as well as a human brain tissue. Finally, we will hear from Christopher Kwan. He will say a few words about the logistics that are involved in brain donation. Chris is our fabulous clinical research coordinator here at our center and also the main contact person for the program. Now, after these presentations, we will have a panel discussion during which we will answer any question you may have. Again, you can post your questions in the Q&A box throughout the webinar, and we will try to get to as many as we can in the second half of this webinar. So the Q&A will be moderated by Karen Gunderson, who trained as a physician assistant and currently runs her own consulting company. Karen has been personally impacted by CAA, which runs in her family, and has experienced firsthand the process involved in brain donation of a loved one. And she is now actively involved in patient advocacy here in the US. And we couldn't be happier that she accepted our invitation to moderate our Q&A for today's webinar. So without further ado, I would like to welcome our first speaker, Dr. Steven Greenberg.
Yeah, thanks very much, Susanna. And it's a pleasure to serve with my fellow panelists and a real honor to speak to this group. Looking through the names, I see many of our patients and research participants who I know personally, and a lot of whom have basically should get their MDs in CAA because they've learned so much about the disease, often more than you ever wanted to. I'm going to give just a really big picture overview of why brain donation itself occupies such a really important part of our research program, and try to motivate that a little bit, and then turn it over especially to Matthew and Susanna to talk about specific research findings, and to Christopher to talk about the nuts and bolts of brain donation. So broadly speaking, stroke is complicated. I mean, kind of everybody knows what a heart attack is, but a lot of very educated people don't exactly know what a stroke is. But I think there are a lot of ways of kind of classifying strokes, but one useful way is have this one big category of symptomatic ischemic stroke. So the ischemic part means that many people know a blockage of blood flow, so plugging up of a blood vessel that supplies part of the brain, and does it in a symptomatic way, so a big enough area of stroke to cause, as is shown on the scans here, that and Susanna's kindly outlining where the area of damage is that kind of sticks out as being different from what you see in the rest of the brain, but not bleeding in this case, it's from loss of blood flow. So that's a big category. And then if you go to the next slide, Susanna, then the symptomatic bleeding strokes that we're particularly familiar with in the CAA field, because that is very often the way that CAA patients first come to medical attention. And this isn't from blocking a blood vessel, but from a blood vessel breaking open and allowing blood to leak out into the brain, often at fairly high pressure, and damage the area around it. And so that white area you see in the CAT scan on the left, that's shouldn't be there. That's blood that's leaked out into the brain and is damaging that part of the brain, causing symptoms. So that's two broad areas. And then a third area that's really important. You might say, well, it's not that pretty small, it's not that big a deal, are these asymptomatic or silent strokes that can be either the ischemic kind, so that white dot where the arrow is pointing on the left, or the bleeding kind, those black dots that you see on the right where the black arrows are pointing. And in both cases, you'd say, well, that's not too bad, but we now know that those asymptomatic strokes really are not so silent, that they often will add up to cognitive impairment in the brain. So you can think of this as being one way to classify three different kinds of stroke. And if you go to the next slide, I then make the point that sort of 2.5 of them are from small vessel disease. So many of the number one, many of the symptomatic ischemic strokes come from other causes, but many also come from disease of this network of small vessels. And you'll hear more about that in the following talks. And then essentially all of number two and the majority of number three all come from disease of this tremendous network of small vessels that feed the brain. So this is often not what doctors think of as being they think of hardening of the big arteries, but it's really these small vessels that determine brain health in a very real way. And so I often say, to think big, you really need to think small for the impact of blood vessel disease on the brain. So go to the next slide. Well, the good news is there are really only two kinds of common small vessel diseases of aging. That number one, arteriolosclerosis, it goes by different names, but it's kind of hardening of the small blood vessels. And it has the risk factors that you think of with hardening of the arteries: high blood pressure, diabetes, and advancing age. And that's one very common form of small vessel disease, but another that's, if not as common, pretty close, is the cerebral amyloid angiopathy, which of course is what brings us all together. And there's only one known risk factor that's typically assessed, which is age, and not a modifiable risk factor. So we kind of know about treating high blood pressure and diabetes. Those things are probably good to help people with amyloid angiopathy, but they're not the fundamental drivers of the disease the way they are for arteriolosclerosis. And of course, our goal is to come up with other things that we can treat for amyloid angiopathy to be able to make a big difference in its disease course. So go to the next slide. Make the point here that the amyloid in amyloid angiopathy is basically the same amyloid that causes Alzheimer's disease. So this is amyloid staining in the brain of a patient who has both and the brown is the positive signal. And so this would be somebody who has both a CAA and these plaques that are the hallmark of Alzheimer's disease. And one point to make here is that this amyloid is I mentioned it's a kind of a protein buildup or a fragment of a protein, and it's made by the brain itself. Most of us think that it's a garbage protein that's meant to be taken out with the brain's garbage, and that instead will build up in the walls of blood vessels or in the brain itself to trigger Alzheimer's disease and amyloid angiopathy. So it's nothing that you eat. It's nothing. Oh, if I stay away from protein, it's stuff made by the body itself. And again, most of us think that there's still some question, but most of us think it's not an important product and it's meant just to be thrown away from the brain, and it lies at the basis of these two diseases of the brain associated with age. So please go to the next slide. And so, our goals for, broadly speaking, across our research program in CA is to identify the pathways that link abnormal small vessels to vessel dysfunction, brain injury. So to figure out what it is that causes a small vessel to malfunction in a way that can either cause the ischemic kinds of strokes, loss of blood flow, or break open and cause the bleeding kind of brain injury, and then to identify treatments that will block these key early steps. So it's as simple as that. Find the key vulnerable points in the pathway where we can do something to slow or stop the diseases. So let's go to the next slide. And it all depends on being able to detect the disease in living patients. And this has been a really big part of our amyloid angiopathy research program, is that, over in the people used to call amyloid angiopathy a disease of the pathologist. The only person who would make the diagnosis would be people like Matthew, who would see the brain after death. But of course, that's not when we want to diagnose something. We want to diagnose it during life so we can figure out what to do about it. And so we've now gone to build a bridge. I put up one of my favorite bridges in Cambridge for any locals, the Weeks Bridge from Cambridge to Boston. And it's a bridge, in this case, from the small blood vessels seen at autopsy to the MRI scans that can be seen during life. MRI gives us that window into the brain. I'll just mention here, this will be coming over and over. Everybody knows we're on a call about brain donation, and it's when we're talking about brain tissue, it's kind of gruesome. This is sort of a topic that can put you off, and we will tread gently. We've all been in the medical world long enough that this doesn't put us off anymore, but we understand that this is a hard thing to talk about. And of course, the people who come onto this call, you really voted with your feet to be part of the research and understand that this brain tissue is really the key to understanding diseases. And the bridge in this case goes in the direction of from the small vessels to the patients. But we want to build the bridge the other direction as well. So we start out with this ability to use MRI to see these kinds of lesions, microbleeds, microinfarcts, the organization of the brain wiring, the function of blood vessels, and deposits of amyloid. So these are the tools that we use in living patients to be able to say what's actually going on at the level of the vessel. And people who participate in research know that know these techniques from the inside, from having done these kinds of scans. And this is what we're looking for when we do these scans, are all these parameters about what's going on at the level of the small vessel. But go to the next slide. And I just, I'll say that one of the biggest advances here was these probable CA. A lot of my patients have heard me and probably heard other doctors talk about when we say probable CA, it means that we're making a diagnosis that used to be made only by looking at the brain under a microscope based on the MRI. And we see this pattern, and this gets into the details, multiple strictly lobar, so it's the location and the number of these microbleeds or this siderosis on the surface of the brain without any other cause. And when we see that, we know with pretty high likelihood that we've got the right diagnosis, that we use that term probable CA, and we know that that's a very strong probability when we make that statement. So you can imagine it's been an enormous tool to be able to lean on to be able to make the diagnosis in people while they're alive and be able to both make decisions and hopefully move towards treatments that will help patients while they're alive, while there's something that we can do about it. But if you go to the next slide, so our goal is to build the bridge really in the other direction, which is understanding what the brain scans are showing and how they got there. So building the bridge in the other direction. That's really going to be the subject of the talks by Dr. Van Velu to what it means to build backwards from the scan, to know exactly what it is that we're looking at, and be able to base eventually moving to treatments on that knowledge. So I think that's my last slide, which means I'm going to turn it over to Matthew. And again, I'll say a few words at the end, but really grateful to everybody who's joined us for this call.
All right, well, thank you, Steve, for the introduction, and Suzanne for inviting me to participate in this. I want to start by saying a little bit about myself and what I do. So I'm a neuropathologist, and one of the things I do is I run a very large brain banking tissue repository, mostly focused on neurodegenerative disease, but I've been collaborating with Dr. Greenberg and Dr. Van Velu for many years now, really focusing and building this effort for CAA. And as you'll hear after me, it's really begun to pay off in terms of what we are able to learn experimentally. But I do want to highlight that we do this by doing an autopsy, examining the brain. And that also does generate a report back to family and physicians that really highlights a deeper level of understanding than is ever possible about what was going on during the course of someone's illness. And I think that really is an additional benefit in beyond the sort of awareness of contribution to research, which is really enormous, and you'll hear how important that is. So I just wanted to step back a little bit and just talk about why blood matters to the brain. So the brain really is a pig. It is massively demanding of both oxygen, what comes in through your lungs, and glucose in order to function. It basically uses about 20% of your body's glucose while only taking up about 3% of your body's volume and mass. So it is basically eating all the time, and getting that oxygen and glucose there really depends on blood flow. So blood flow is pumped by the heart. It has to be oxygenated by the lungs, and it has to have glucose in it from your eating and your liver and the rest of your body that give that nutrition to it. And that's what lets the brain function, both function normally and unfortunately function abnormally. Just a little terminology, the blood comes to the brain by arteries, and it starts off in very big ones, and they get smaller and smaller as we go. And it then has to leave the brain by veins. And people may be aware of all of the press that was around the Johnson & Johnson COVID vaccine and risk of stroke mostly in young women who got the vaccine. And that was really related not to any of the input, but to blockage of the output. So this balance of in and out is really important. And the capillary is the very smallest branch of the vessel. And that's where oxygen leaves the blood supply and glucose leaves the blood supply and get out into the brain tissue itself in order to function. So Suzanne, if we go to the next slide. So the big vessels that feed up to the brain are your carotid arteries in your neck. And those are shown down there with the little label ICA. And they will branch and branch further and branch further and run all over the surface of your brain. But eventually they penetrate in and deliver blood to that sort of orangish ribbon on the surface, which is the cortex. That's where the nerve cells are that do the thinking that you're aware of, the controlling of movement, the awareness of sensation, your decision-making process, and so on. But if we go to the next slide, and this is about 1 millimeter across the top and 1 millimeter down the side and about 0.5 millimeter deep. And we're basically walking through that. And what's highlighted in white are all the blood vessels. So no part of the brain is more than 1/10 of a millimeter away from a blood vessel ever. So this is a massively complicated network that's present all over throughout your brain. And what we're trying to understand is what happens when one of those little branches has something wrong with it. And that's really the issue that requires the very sophisticated work that you'll hear about next from Suzanne to really find them. So if we go to the next slide, if we look at just a very, very thin section of this that we look at under the microscope, and this is magnified about 200 times as I show it to you here, there are some vessels, and I tried to get a vessel running right down the middle that Suzanne's going to point out there. There's a vessel. There are other ones nearby, sort of down a little. Yeah, they're down there. And then if you just go over and then over, they're evenly spaced. Those are the blood vessels that we're talking about. And the cells in between them are the nerve cells that are actually doing the thinking for you. And this is a piece of normal brain. And if we go to the next slide, this is an abnormal, this is someone with bad CAA. And if you look at those vessels, you can just see how thick they are. Compare them to how thin the wall is in the vessels from the person without disease up there. And the other thing you can see is that the cells that are normally there are gone. So the blood vessels normally have a type of muscle, have a muscle cell in their wall that allow it to open up and get tighter. And those cells vanish, they die when the amyloid deposits there. And so all of the things that are critical for understanding why someone has a bleed come from what happens to, what makes the amyloid appear there, what makes it kill the cells, and then what makes it break, what allows it to make that vessel bleed. If we go to the next slide, this is actually, I mean, we've all had the experience of banging our shin on something and you end up with this black and blue mark. This is essentially the tiniest black and blue mark on the surface of the brain. Surface of the brain is right up at the top of the picture, okay? And then as Suzanne is highlighting, right in the middle is the vessel that broke. And the brown around it, the brain can't clear the black and blue mark as well as your skin does. And so it remains. And when you see them, when Dr. Greenberg and others, or Suzanne Van Villa, when they see them on imaging, what they're seeing is that area around it, because that shows up very, very strongly on the MR scan. So what we really want to understand by finding these lesions, and finding them is hard when you are confronted with a brain. It's a big place, and this is tiny. That's 0.5 millimeter in diameter. And it's a sort of sphere, so we have to be able to find that. And really the critical piece of the work that's been going on is being able to use the tissue to find them so that we can then really study their anatomy. What characterizes the vessel that bled? What's going on in the brain around it? What's the type of reactions that happened? And those are really the critical questions there. If we go to the next slide. I think the other thing I'd like to say is that over the last 15 years in collaboration with Dr. Greenberg, we've really described not just the bleeding that comes with amyloid angiopathy, but the other types of reactions. So some people develop sort of an antibody, an immune reaction to the deposits in their brain. And while that goes on and can damage the vessels further, it can also lead to other problems. And through studying patients and biopsies from them and some autopsies, we've really come to understand that process as well. So that it is a very complex and clearly dynamic disease that in different areas of the brain is at different stages. So really being able to examine whole brains for research really makes an enormous impact on the work. And I just want to close, I think, as Suzanne said at the beginning, this is an enormously difficult decision for families, but I think it has the ability to both feel as though one is making a contribution to diseases that are really otherwise sort of still, they're better understood, but we don't have that much we can do about them. And I think that that's the goal, is not just to be able to diagnose, but to be able to think about how to treat them. All tissue, and Christopher will talk about this later, all of the donations, everything is used in a de-identified manner so that, while we would be happy to share the results, we don't say, who contributed, which brain contributed, which piece of tissue for which study. That's all done in a completely protected manner. And that's sort of the critical most issue for us, one of our responsibilities, in addition to making sure the tissue is available for research, is to make sure that it's available in a completely de-identified manner, so that it's, you know, a contribution that is, in that sense, you know, clean. And finally, I'd say that to the extent that there are genetic factors that play into CAA, we can use donated tissue to support that. As we do it as a research activity, it's not necessarily reportable back, but because we've also handled all of these autopsies as clinical cases, we are able to report clinical information back in that manner. So there is ongoing potential value to families that can come from that. And with that, I'll stop. I know that Suzanne's going to circulate email addresses later, but I'm happy to answer questions offline from others who may want to ask them at that point. So I'll stop there.
Thank you. Thank you so much. So it is my pleasure to really share with you a little bit more about what we've been able to do with some of these precious brain donations. So since the start of the CAA research program, Brain Donation, about a little over 5 years ago now, we have received approximately 30 donations so far. And this has really allowed us to get a unique perspective into the brain of someone who suffered from CAA. So as Steve Greenberg just mentioned, the diagnosis is made on the basis of bleeds on an MRI scan, as you can see here on the left-hand side. However, these bleeds are only very infrequently found at autopsy because neuropathologists such as Dr. Farsh, they cannot simply examine the entire brain under the microscope. So therefore, for a long time, we really did not have a complete understanding of what, first of all, one of those microbleeds look like under the microscope, but more importantly, what actually caused the microbleed in the first place. So what these brain donations have allowed us to do is to really scan someone's brain after they have passed away. And as you can see on an example, in the next slide of what one of those brain scans look like. You can see from this image that we just use one half of the brain, but we can scan it at a very high level of detail. And there are really two main reasons why we do this. And the first one is that we can take a much better picture of the brain this way. For those of you who've had an MRI scan, you know that it's not really easy to lie still in the scanner for more than an hour. So we have to make rather quick snapshots of the brain during life. And sometimes these turn out blurry because patients tend to move their heads in the scanner. But scanning a brain after autopsy, like here's an example, we can really leave these brains in the scanner for much, much longer, often 12 hours, and sometimes even up to 24 hours in very specialized MRI scanners. Now you can imagine that by doing this, we can take much better pictures as shown here. Now the second reason that we do this is that we can then cut the brain in our lab and find the same exact bleeds that we've also seen on an MRI scan during life, and then examine that specific bleed under the microscope to find the blood vessels and to examine what caused these vessels to break in the first place. So in our studies, by really carefully examining the brain scans that were made during life with those that are made after autopsy, we can really establish when some of these bleeds have happened. And what you're looking at here is just a cross-section of a brain scan shown here. And this is the corresponding level of the brain after it has been cut. So we then take a few samples, and this is just one example here of certain locations of interest, and we process these tissue samples in the lab, and this is shown here. There's a sample located in a standard tissue cassette. Here again, you can see the same area. I've now flipped it because it's easier to compare to the image here below. And you can see that this area here has undergone processing in the lab, and the tissue is stained with a certain dye so that we can highlight the individual brain cells and the blood vessels of the brain. And this is just a digitized image of one of those stained sections. And on our computers, we can then zoom in all the way to examine the tissue, just like Dr. Farsh showed an example just a few slides ago. In this particular area here, we have zoomed in and stained the tissue with a different method to really highlight the amyloid that is accumulated in the blood vessels. So what you see there is every brown stripe in this image is a blood vessel that has CAA in the wall. Now we've taken this approach one step further and scanned even smaller brain samples. And this is just a couple inches wide by an inch thick, and to obtain even better quality images. The two black dots that you see here are two examples of small bleeds in this particular brain tissue sample. And you can see these now in much greater detail on this very clear picture of the brain that we've acquired with the specialized MRI scanner. Now, what we then do in the lab is that we process the entire tissue sample, and by cutting them in ultra thin sections, and that is what is shown here. These are just a few of these sections sort of lined up that are all successive very thin sections throughout the entire block. And the reason we do this is that we can then find every individual bleed in this tissue sample and also the individual blood vessel that has led to this bleed, that was responsible for that bleed. And using this approach, which you can imagine is quite labor intensive, we have the really unique opportunity to study these blood vessels and to figure out why they bled when the patient was still alive. So on the next slide, you can see an example of what such a bleed looks like under the microscope. So now we're zoomed in pretty far. You can see at the scale bar here. This is 500 microns, so just 0.5 millimeter. And this is what the bleed looks like under the microscope. So all these pink dots that you see here in the brain tissue, those are really the blood that has leaked out of the blood vessel into the tissue of the brain. And what you can see here is the individual blood vessel that has bled. The fact that it's yellow, that indicates that this is the source of the bleed. Again, this is the surface of the brain where you see the larger blood vessels. So we really have now the opportunity to study those blood vessels and to figure out what happened to these before this vessel bled. So in this next picture, you see again a stain for amyloid of the same blood vessel. So this is just an adjacent thin section of the same area. Now, after carefully examining many of these types of vessels in these brain tissue samples, we've been able to figure out a little bit better what may have happened before these vessels bled. And we're now beginning to sort of understand that just the severe buildup of amyloid in the blood vessels, it initiates this cascade of events, which makes, first of all, the blood vessels leaky, and the tissue surrounding the blood vessels kind of inflamed. There's like a local inflammation going on in the surrounding tissue. And this process altogether seems to trigger removal of amyloid from those vessels, which then initiates bleeding. As you can see here, this part of the blood vessel is actually less brown than this part here. So we know that it happened here and that all these events took place before these vessels bled. Now, observations like this really help us to better understand the biology of the disease and also help us to think about new intervention strategies. Because the next step will be to figure out in the lab with experimental models how we can stop some of these events from happening and hopefully in the future prevent bleeding in patients with CAA altogether. So the next slide, just to summarize, so far, based on these brain donations over the last 5 years, we've been able to publish a total of 7 scientific articles as a result of these brain donations. And we're currently working on four more papers to be published, hopefully in the near future. And here's just that same list that the Dr. Stephen Greenberg just showed and mentioned earlier, that we've actually been able to study under the microscope using these brains. So really, thanks to donations like these, it really allows us to do the research that we do in the lab. So thank you very much. And with that, I would like to give the floor to Chris.
Hi, everyone. My name is Christopher Kwan. I'm a clinical research coordinator here at the MGH Hemorrhagic Stroke Research Center. I'll be the main point of contact for coordinating our brain donation program, and I'll be happy to take you through the overall process for not just enrolling in our program, but what you could expect once you have enrolled. So we are honored and grateful to be considered for every brain donation. To make this process as smooth as possible, there are many logistics that must be considered. When you or your family member are interested in enrolling in our brain donation program, I will be in contact with you to discuss the process in much more detail. Please just remember that the decision of whether we can accept the brain of an interested donor is made by Dr. Greenberg. If, say, an interested donor has never been seen at Mass General Hospital, we would appreciate as much neurology-related medical records as possible to help make that decision. And in particular, any previous brain imaging would be the most helpful. So once Dr. Greenberg has given me the green light to go ahead, the donor and or the family member will sign the intent to donate form, which is a consent process. We will work very closely with you to accommodate any specific plans, including funeral arrangements and respecting any religious or cultural customs. For your information right now, our donors can be cremated and have an open casket funeral. And if you have consented before a donor passes away, I'll be in touch with the family members to discuss and arrange as much detail as possible in advance. So at the start of a donor's significant decline, we ask that a family member or a legal representative initiate contact with me and the pathology department at your nearest hospital to arrange plans. Please work with us to find a hospital or an academic medical center that can perform the autopsy and obtain proper consent if you're more than 200 miles away from Mass General Hospital. It's really important that the earlier this can be done, the better it is for the whole process. And I'll assist in arranging for transportation to and from the donation site where the autopsy can be performed. Now, donated brain tissue will be stored and examined carefully by the MGH pathology team led by Dr. Frosch. And just like Dr. Frosch mentioned, any contribution to the brain donation program will be de-identified to protect our donors' privacy. After your donation has been received, we'll be in touch by phone or a letter regarding the information and instructions on how to receive the autopsy results through the Mass General medical records. And as a final note, for all of these steps, I'll be available to answer any questions or concerns. I know that these steps can occur under many challenging circumstances, but we'll do everything we can to assist you through the entire process. Thank you.
Let me just stop share and then we can transition to the moderated Q&A part of the webinar.
Excellent, thank you. So I'm very honored to moderate and be involved with this CAA webinar. As previously mentioned, I've had 2 family members who've donated brains to the MGH program, and I'm very excited to be here and help out with some questions. So without further delay, we have some questions in the chat as well as some that came in through the registration process, and we'll try and get through as many as possible as we close this talk. So I think to start out with, when we talk about brain donation research, this is for you, Dr. Frosch. Can you discuss what type of information is provided to the family, and if there's specific research, if the family can figure out what papers are actually published regarding their family members?
Sure. The first thing is that the autopsy report is generated, and then that's available to the family. It's slightly complicated to get the actual final copy, but we do communicate usually back through the physician who was the point of contact, either the treating physician elsewhere or Dr. Greenberg. And I often will speak also with families who are interested in understanding what was found. So the information definitely gets back to the family about the exact diagnostic findings. The communication of research, as I said, we don't identify which brain donations go into which particular manuscript. Everything there at that level is done in a completely de-identified manner. But what we are doing, and I know that there's a newsletter that comes out from the group in the Hemorrhagic Stroke Center, which does communicate research activities. There are mailing lists and the like that bring people up to date with recent publications that will have drawn from the bank. But part of our obligation as the holders in the bank is that we make a barrier between anything that is identifiable about the donor. That information is only shared back to the physician and family. And then with the researchers, it is always de-identified in that manner.
Thank you very much for that. Chris, you mentioned a little bit about logistics and how far away that families and patients can decide to donate. Can you discuss if people are further away, how you go about doing that?
Sure. Every brain donation is unique in that obviously there are many moving parts, and logistics, especially when it comes to transportation, can be very tricky. So if an interested donor is located within 200 miles of Boston, our MGH pathology team can perform the autopsy. So that includes much of New Hampshire, Connecticut, some parts of upstate New York, Maine, some parts of Maine, as well as Rhode Island. However, if an interested donor is further away than that, it's very important that the family identify and contact their nearest hospital or an academic medical center's pathology department. And we'll work with you to find one that can not only perform the autopsy itself, but also obtain the proper consent for brain donation specific to our program's requirements. So it's very important for you to get in touch with us.
Great, thank you. There were a couple questions about genetic studies and whether or not CAA is hereditary. So Dr. Frosch, you had mentioned that there were some genetic studies tested during the autopsy process. Can you go over that?
Sure. So, so there are some relatively uncommon across the broad population of individuals with CAA forms that are genetically determined. These are changes in the sequence of the structure of the protein that gives rise to the amyloid itself. And those seem to put individuals at risk, and those are inherited one generation to the next. And the sort of best well-studied group like that is the large, relatively large by comparison cohort of the sort of Dutch CAA kindreds, and both Suzanne and Steve can speak to those in far more detail than I. We can test if that is a question. The tissue that's collected at autopsy can be used for testing for that. We do that by working with a genetic counselor, because if a family is going to get a new diagnosis of a heritable disease, that has implications for everyone in that family. And so we don't do that lightly. And there are genetic counselors who work with us who are experienced in these sorts of issues. And they sort of make sure that the family understands what they're getting from that, especially in the face of a disease without current treatments. But also that not everybody in a family might want to know what their individual status is, and can be very complicated. And so we rely on their expertise to help us sort of negotiate and navigate those waters. But that really is the sort of those familial forms are a relatively small fraction of the broad population who is affected by the disease. It does affect some, and it's clearly an important part for those families. But sort of across the board, it is a relatively less common, unlike some diseases like Huntington disease, where essentially everyone who has it is part of a family that has it.
Thank you for that explanation. Since we mentioned the Dutch variant of CAA and hereditary, Dr. Greenberg, can you discuss where else in the world there are patients with CAA besides the Netherlands, and how brain donation at MGH and that research carries over to the global community?
Yeah, thanks very much, Karen. This is a truly global problem. So the Dutch take a certain pride in having such a prominent role, and of course, we're delighted to have Susanna, for example, come and join us from her home country, the Netherlands. But it's a truly global disease, and there's probably nobody who's not at risk. That there are. It's a funny thing. CAA. I don't get this question much anymore. I used to get the question of, is this a new disease? I didn't think anybody ever got this, and a lot of it was what I mentioned early on, that it used to be only diagnosed to people who had come to autopsy, and the fact that we can diagnose it during life has suddenly opened up many more people who have it. And also, there's another thing going on, which is everyone is living longer. We've done a better job of preventing heart attacks and cancers and things. I mean, those, still many people die from them. People live longer than they ever did, and aren't so much dying from other things. And even, when I mentioned that arteriolosclerosis being the competing small vessel disease, the better job we do of treating high blood pressure, for instance, the larger proportion of bleeding strokes that are being caused by CAA, because it's not that CAA is becoming more common, it's that other things are becoming less common. And so especially in Western countries that have had more success at treating high blood pressure, CAA has taken on a really big role, and that's certainly international among richer countries, and it still plays a very substantial role even in less rich countries where high blood pressure still is not well treated. But there aren't any populations, there aren't any racial groups, any countries or ethnic groups that are really protected from CA. So any place I go, I hear about and see patients who have it. The focus on these hereditary forms has been the possibility of doing the kinds of very early research studies that can't be done in people where we don't know about their CA until they're already having symptoms. So there is a special place in the families and the countries that have hereditary forms to be able to learn about the very early steps. But that doesn't, and that means that there's this International CA Association that's based in the Netherlands, I think, because of the historical interest in these hereditary forms. But it's truly an international disease and has international attention and has been on the radar screens of neurologists everywhere. This is not a small problem, and it's not even a rare problem. It's really a pretty common disease as we now understand.
Very good. Thank you for that answer. While we're talking about research, Dr. Van Velu, can you discuss other examples or new findings that have come out of the brain donations of people with CAA that have donated to your program?
Yeah, happy to. So I gave just a small example because of limited time of what we've learned from small bleeds in the brain by looking under the microscope. Another study that we did recently is looking at the other type of stroke, that is the microinfarcts, and that we see often in the brains of patients with CAA. And these are really hard to see really during life on MRI scans because they don't show up the same way as the bleeds do, and our techniques are not as sensitive to detect these smaller types of stroke. But we are, in fact, have understood from large autopsy studies in the past that they are very common in the brains of older patients with both Alzheimer's disease but also CAA. So doing these types of studies, we've actually been able to study them in a lot more detail, and being able to identify them, sort of the MRI signatures, if you like, of how to identify them during life. So that is almost taking the opposite approach of what I just showed you, of going from an MRI scan to pathology, because we actually took the opposite approach, going from pathology back to MRI to figure out what these tiny lesions look like on a scan. This is another example of what we've been able to do.
Excellent, thank you. There's a lot of interest, Chris, about studies and any type of clinical trials going on. Can you discuss the difference between an observational study and a clinical trial, and what is currently going on at MGH?
Sure, I'll be happy to discuss what's currently undergoing from our center. We are currently enrolling patients in several studies, which we hope will advance our understanding, diagnosis, and treatment of CAA. All of our studies take place out of Mass General Hospital in Boston, and participants must be seen clinically by one of our neurologists, including Dr. Greenberg, to be eligible to participate in our research studies. As a note, we are not currently conducting any drug or treatment trials. So though we have in the past and will likely in the future, many of our patients who had enrolled in our imaging studies have either participated in or shown interest in the brain donation program. So when these patients make the decision for brain donation, we know that, like Dr. Greenberg mentioned, they complete this chain of research data that provides the final link between a participant's research scans and the microscopic examination of his or her brain tissue. So that kind of unique ability to examine the brain microscopically is often the missing link in our research studies that are undergoing. So in many ways, the brain donation program really is the greatest contribution a patient or their family or a research participant can make towards aiding this fight against CAA.
Thank you very much. So in the event that a patient hasn't been seen at MGH and hasn't participated in a clinical study or observational study through MGH, Dr. Frosch, can you discuss what type of medical records are needed then and what's important in the brain donation process?
Sure. I was actually just answering a similar question online when I was typing in the background. So as Dr. Greenberg pointed out, the goal is to have that bridge between what we can do with a brain that we have collected, both the incredible imaging and what we do under the microscope, and really bridge that back to scans that people get when they go to see their doctor or as part of a research study. So having those scans, the data and the actual scan, not the report, but the scan itself, to hold one up against the other is really valuable. So that sort of information is very important. Knowing about episodes where people had symptomatic events, they had what was a bleed and they ended up in the hospital, those sorts of things, and really sort of visits with the neurologist who was caring for their CAA and whatever other neurologic diseases. All of that information is really the critical one, but I'd say that the most valuable is having copies of the disc that the scans are on, just so that we can really make sure that each end of that bridge is well anchored on the bank of, the uncertainty in between them.
Great, thank you. I don't think we could end Q&A without asking about treatments and what's on the horizon. So Dr. Greenberg, if I can ask you, can you tell us what's on the horizon for CAA and potential treatments that may be promising?
Yeah, I mean, I think thanks, Karen, as everybody knows, I put CAA in the group of neurodegenerative diseases. It's a blood vessel disease, but it shares really a lot of characteristics with other diseases that you read about and all the time, Alzheimer's disease and ALS and Parkinson's disease that we put in, Huntington's disease, we put in this category of degeneration, something that where in this case blood vessels little by little suffer damage over time. And the kind of holy grail for us in neurology is to have what we call disease modifying treatments. We're happy when we can help patients in any way, but what we really want to do is slow or stop the disease from progressing before it gets to a point that it's debilitating to people. And that's been really hard. We always thought if we just understand all the steps, it won't be hard to come up with treatments, but it's actually been very hard. I feel like, when you're a kid and you're sitting in the back seat of the car and you ask your parents, are we almost there? And they say, oh, it's right around the corner. And you go around the corner and you're not there. And you say, well, are we almost there? And they say, yeah, it's just around this next corner. And it just keeps going on. And it's frustrating for everybody on this call that we want progress and we want it now. I've never been more optimistic. We have a lot of potential places to intervene. I've mentioned that we need to understand the key steps, and I think our understanding is really unprecedented in knowing, well, if we could stop this step, we could stop this one. The kinds of things that Dr. Monvelu, Dr. Frosch outlined in terms of preserving blood vessel function, preventing damage to the blood vessels, these different steps that go into causing bleeding, causing loss of circulation. And but each getting from there to results in animal studies to moving to human studies, is really hard. It's even when you have the right plan, each clinical trial is kind of like building a new ocean liner, like building the QE2 to cross the Atlantic. It's just it takes a lot of resource, a lot of money, a lot of scientific support, and a lot of steps to show that your treatment is safe, that can be given in people, can be given in people in the doses required. So nothing comes as fast as we want it to. We're as impatient as anybody on this, but I'm extraordinarily optimistic that we will have disease-modifying treatments. We're going to have them before we quit doing this stuff. This has been our goal all along, and it's tantalizing, but nothing will come overnight. So everyone, I think, on this call has voted with their feet to be part of research and to help make that happen. And all of us as panelists have committed ourselves to be part of that and make that happen. So I'm optimistic, but you have to be chastened by how hard this road has been and know that it won't be easy and it won't happen tomorrow, but it will happen. I have no doubt.
Thank you. That's an excellent perspective. So with that, we're at time with the question and answers. I know there's quite a few that haven't been answered in the chat. I thank everybody for participating and for submitting their questions. We'll definitely take note of this for future calls. I'd like to thank my fellow panelists for your expertise and for your time and attendance. And with that, and to finish the evening, I'd like to turn it over to Dr. Greenberg once again for final thoughts.
Well, I just kind of gave them, but it's sad to say again. I mean, what an honor it is to work in this community, and both and when I say this community, I mean the scientists in the community, the healthcare professionals, and the patients, the participants. There's that the contributions that people make of their time, their money when people are able to raise money or donate, and no contribution is greater than brain donation. I mean, it's the most personal. Susanna and Matthew both alluded to this. It's the most personal contribution that anyone, a family member, a person can make. To say I'm so committed to this that I want to donate myself to it. It's just if we ever had any doubts about the importance of what we're doing and how committed the patients and families are to beating this disease, that's the kind of thing that reminds us, not that we've had any doubts. So it's a pleasure to speak with this group and to continue to work with you going forward.
Thank you very much.
Bye, everybody. Great to be with you.
Thank you all.
Yeah. Thank you so much. I will stop the recording.